Okay, so it is my pleasure to introduce to you Dr. Hopi Hoekstra. So Dr. Hoekstra received a BA from the University of California, Berkeley, after which she pursued a PhD in integrative zoology at the University of Washington and a postdoc at the University of Arizona. She took her first faculty position at UC San Diego in 2003, but in 2007 moved to Harvard University, where she continues to do her research today. Dr. Hoekstra has received numerous awards and recognition over the course of her career, including election to the National Academy of Sciences in 2016. And currently, Dr. Hoekstra is an HHMI investigator and an endowed professor of organismic and evolutionary biology and molecular and cellular biology. So Dr. Hoekstra's research focuses on the genetic factors that underlie complex traits and the molecular basis of evolutionary change. And we're so delighted to have you here today, Dr. Hoekstra, to talk about your work. So please, everyone, join me in welcoming her.
So first, I want to thank you very much for the invitation to be here and the lovely introduction. It's so fun to be giving seminars in person once again. Today, I'm going to tell you a tale of tales. So before I begin, I want to thank the folks who did the work that I'm largely going to talk about today. And this includes two former graduate students, Evan Kingsley and Emily Hager, who both recently graduated and have taken on new postdoc positions. So throughout the talk, when I say we, what I really mean is Evan and or Emily. So I don't need to tell this group that there's a lot of diversity in body segments among animals, and they can vary on multiple axes. So for example, they can vary in size, as illustrated by these two annelids. They can vary in identity, and of course, they can vary in number. But they can also vary tremendously even within smaller taxonomic units. So in mammals, for example, the vertebral column has emerged as a model system for understanding the evolution of segmentation. And that's because the vertebral column evolves in some really interesting ways. As many of you probably know, the cervical or neck vertebrae in mammals are largely conserved. So with a few notable exceptions, all mammals have 7 cervical vertebrae. The thoracic and lumbar and sacral segments also expand and contract both in absolute terms, but also relative to each other. But really all of the exciting evolutionary action really occurs at the tail end, the caudal vertebrae. And these sort of patterns of evolution are nicely illustrated by these images here. So we have a giraffe, a primate, and a rodent. And despite having very different neck lengths, they all have 7 cervical vertebrae, so that's all changes in length of vertebrae, whereas the tail, for example, vary in both size and number of vertebrae. So you may wonder, why is the tail so diverse? And this is likely linked to its function and its multiple functions and uses by different mammals. So for example, tails can be used as rudders in flight, for propulsion during swimming, for balance in sartorial bipedal rodents. It can even be used behaviorally as a sunshade. And probably most dramatically, it can be used as a 5th gripping appendage in those prehensile tailed species. And this is relative to humans, of course, which are little nubby, quite boring tails. So as an evolutionary geneticist, when I'm faced with all this kind of tremendous diversity in tails, one of the first things we like to do, or one of the major approaches we take in my lab, is to ask, what are the genetic changes that are responsible for this variation in this case, tail length? Now, for many geneticists, identifying the genes or the mutations in those genes represents an end game, sort of finding the proverbial needle in the genomic haystack. But for us, this really represents the beginning, because once we identify a gene and or mutation in that gene, we can use that as a handle to ask questions about process and mechanism. And in particular, we're interested in 2 types. That is the evolutionary processes that are driving variation in the trait of interest, but also the molecular mechanisms that give rise through changes in genes, through development to produce that variation. So in other words, we want to know both why traits vary and how they vary. So today I'm going to focus on one particular species. So let me introduce you to my favorite organism. This is the deer mouse, Paramiscus maniculatus. It's the most abundant and widespread mammal in North America. and it's been the subject of studies by natural historians for over a century. Most notably by a fellow named Wilfred Osgood, who in the early 1900s traveled around North America, catching mice and describing variation both within and between populations. And this resulted in a taxonomic revision, a giant book, in which there are many notable patterns, but one of the most striking patterns that he saw when he examined populations of these particular mice is this: essentially they come in two flavors, a forest form and a prairie form, in other words, ecotypes. And Osgood claimed that if you handed him a mouse, he could look at it, and just by looking at it, he could tell you if you trapped it in the forest or in the prairie. And that's because these mice differ in a number of morphological features, including forest mice having darker pigment, longer whiskers, bigger ears, larger feet, and most notably longer tails. What's remarkable about this divergence is that it likely occurred only over the last few thousand years. As the glaciers retreated at the end of the last ice age, the ancestral prairie form could colonize this novel forested habitat and adapt on multiple morphological traits. And even more remarkable, it's likely that this happened not once, but multiple times. We know through using molecular markers, there's at least two independent evolutions of this novel forest form, one occurring on the West Coast and one occurring on the East Coast. And as an evolutionary biologist, this is really exciting because essentially what this means is we've done two evolutionary experiments on these two coasts. So because of this, it's led us out of the cozy lab environment and out into the field, where we went to Oregon and trapped mice, both forest and prairie mice. And then on the East Coast, again, a forest and prairie population. And we were able to bring those back to Harvard and establish laboratory colonies in which we could study things in nicely controlled environments. So for the rest of the talk, I'm going to focus really on these four populations. Forest data will be color-coded in green, prairie in brown, and the colors from the West Coast will be slightly darker. Okay.
Can you talk about what you know about the mixture across the populations?
Yeah.
I mean, maybe you're going to do that, but do you know?
I'm not going to talk about that, but there's a lot of gene flow. So these are really abundant. The effective population sizes are very large. They have a lot of genetic variation and a lot of it's shared. There is some structure, and one of the most clear patterns, demographic patterns, is there's sort of this West Coast lineage and this East Coast lineage. So within the West Coast, there's a lot of gene flow. Within the East Coast, there's a lot of gene flow, but there's certainly limited gene flow across that boundary. That helps. Yeah. We've now sequenced hundreds of genomes of mice from across the range, so we can answer that question in much more detail now. Okay, so as I mentioned, I'm going to focus today on tails. So the first thing we wanted to do is just ask the question, were the observations that Osgood made a century ago really true? And indeed, that's what we see here. So here we're looking at tail and body length on the vertical axis. And what you can see is that forest mice have tails that are longer than the length of their body compared to prairie mice that have tails that are shorter than the length of their body. The second pattern to note is the magnitude of difference is about the same on the West Coast and the East Coast between forest and prairie forms. And the third thing to note is, for reasons we're still uncertain of, the mice on the West Coast within a habitat type have slightly longer tails than the mice on the East Coast. Just to note that what we're calling forest on the West Coast is not exactly the same as forest on the East Coast, and same with prairie. So these are very large sort of ecological classes. Okay. So clearly the tails differ, and that's correlated with environment. The first question we wanted to know is why? Why do tails differ? And I'm sure you're all thinking, well, this might have something to do with the fact that forest mice live in forests and they have opportunities now to climb. And this is an idea that Osgood, in fact, raised. And so it's something that we wanted to test. So Emily Hager in the lab decided to design a behavioral assay, which involves two platforms that are raised, connected by a narrow rod. She introduced a single mouse into this enclosure and let it freely cross the rod for 5 minutes, videotaping it from multiple angles. Now, while this certainly doesn't capture all the complexity of a mouse climbing in a forest, we do think it captures at least some ecologically relevant features, because these mice clearly cross narrow rods or twigs in their natural environments. So we can use this assay to start to quantitate differences in their behavior. So next, what I want to show you is just two videos. Importantly, they represent the mean behavior for forest and prairie mice. So these are not extremes. You can see that there's two platforms, the narrow rod, and I want you to focus on two things. One, their overall performance, but also how they're using their tails. So first, I'm going to show you an eastern forest mouse. So you can see this guy nicely traverses the rod, but uses its tail both as a stiff counterbalance, but also needs the flexibility to wrap around the rod. And as I mentioned, we can quantitate this now using software. Okay, so that mouse did a pretty good job. This is in stark contrast to a prairie mouse, which although he gets on the rod, he flops over, tries to right himself, again, uses its tail both in a flexible way and in a counterbalance way, and we can quantitate its poor performance. Okay, so using this assay now, we can quantitate differences in their performance. And I'm just going to show you quickly some data to do just that. So as the video suggests, forest mice spend most of their time upright, around 80 to 90%, whereas the poor prairie mice spend the majority of their time hanging upside down, clinging for dear life. And this is for two reasons. One is when a mouse is upright, a forest mouse stays upright for a longer amount of time. And second, if they fall over, a forest mouse can right itself back upright more quickly. And finally, once upright, a forest mouse can move more quickly across the rod. So in other words, what these data suggest is that there's a stark difference in climbing performance between forest mice with long tails and prairie mice with short tails. But remember, these mice differ not just in tail length, but in a number of morphological features. So this just shows an association between tail length and performance. But towards the end of the talk, I'll try to provide you with some data that shows tails actually matter, independent of those other morphological traits. But for now, we're going to turn to another question, and that is how. And we're going to ask this question at 3 levels, starting with morphology. So if we think about a mouse's tail that's comprised of these caudal vertebrae, to get a longer tail, we can think of two sort of very simple alternatives. One, to get a longer tail, you can keep the number of vertebrae the same, but you can just increase the length of individual vertebrae. Alternatively, you can keep the length the same and just add vertebrae to the end. So to ask which of these two approaches or combinations thereof evolution used in this transition from prairie to forest, we took a lot of x-rays, designed a program that allows us to measure in an automated way tail length, count the number of vertebrae, and I'm going to tell you data about the length of the longest vertebrae, but all the vertebrae are highly correlated in terms of length. And in doing this, what we found is that forest mice have both longer and more vertebrae, and this is true for both the east and the west coast. So for here we have longest vertebra, and here we have sacrum length as a measure of body size. So for any given body size, forest mice always have longer vertebrae. But they also have more vertebrae. So for example, on the west coast, a forest mice have approximately 28 on average vertebrae compared to about 24. So next, what we wanted to do is ask, of these two sort of strategies, how much do they contribute to the difference between forest and prairie tail lengths? So in other words, how do you go from a prairie tail to a forest tail? So here's cumulative tail length. We're marching along the tail on this axis, and you get 50% of the way from prairie to a forest tail by adding vertebrae. And you get the other 50% of the way there by elongating vertebrae. And this is true not just on the West Coast, but also on the East Coast. In other words, both of those strategies contribute equally to the difference in tail length. So we were quite struck by the fact that we see this remarkable similarity in morphologies in these two independent replicates. And one of the questions we wondered is are mutations that contribute to a longer tail pleiotropic? In other words, affect both length and number of vertebrae. So to test that hypothesis, what we could do was use genetics. So we did 2 independent crosses, one on the West Coast and one on the East Coast, where we took a short-tailed prairie mouse, crossed it with a long-tailed forest mouse, generated F1 hybrids. These lines represent their chromosomes, notably that have intermediate-like tails. Then we intercrossed these mice to generate a large genetically heterogeneous population of second-generation or F2 hybrids, where each mouse has a different combination of alleles from the prairie and forest parents, and hence have a wide range of different tail lengths. So in these genetically shuffled mice, we could then ask, is there a correlation between the length of vertebrae and the number of vertebrae? And what we see is there's no correlation at all, either on the West Coast or on the East Coast. So this is not driven simply by pleiotropy. But now that we had all of these mice and we measured all of their tails and their component parts, the next thing we could do is ask another question. That is, how do they get longer tails from a genetic perspective? Because with all of these second generation hybrids, we can now genotype them at hundreds of thousands of markers across the genome and ask, are there any correlations in the genome in which genotype is predictive of tail phenotype? In other words, we're using forward genetics for QTL mapping. So when we do that, what we can see when we consider total tail length, there are 6 regions of the genome that show a statistical association with total tail length. So here's our LOD score. You can think of that as our statistical association between genotype and phenotype. Here are our chromosomes, and when that association crosses the genome-wide significant boundary, these gray bars indicate the confidence interval that suggests that there is a mutation or mutations in those regions that affect tail length. So this was kind of interesting to a geneticist because a single gene mutation is a Mendelian trait. It's a little bit boring, but if you have hundreds of genetic changes, that becomes intractable. So it looked like there were about 6 regions, but we could also ask, well, not only what are the regions that affect total tail length, but what about its component parts? And what you can see is that there are 3 regions of the genome that affect length of the longest vertebrae that overlap with these 3 QTL that contribute to total tail length. And in a remarkably symmetrical result, there are 3 other QTL. The other QTL affect the number of vertebrae. And this is interesting for 3 reasons. First, it perfectly explains why we didn't see in those F2 hybrids a correlation between vertebrae length and number, and that's because the causal mutations for those 2 traits lie on independent chromosomes. Second, of the 40% of the variation that are explained by these QTL, 20% are explained by the longest vertebrae QTL, and the other 20% by the number of vertebrae. Again, consistent with that idea that these 2 strategies contribute equally to the difference in tail length. And then third, in each of these QTL, it's always the forest allele associated with longer tails. And that's statistically different from random. And that's, again, evidence that suggests that selection is driving this difference in tail length, with the forest alleles contributing to longer tails. Okay, so these—
Do you have when in development, these 2 phenotypes?
I will get to that, I promise. That's great. Okay, so these are the data from the East Coast cross. What's going on in the West Coast? So here, again, when we did the QTL analysis on the West Coast, we found, again, a handful of QTL, in this case 5. But the question is, do any of these overlap? So when we superimpose the ones from the East Coast onto the West Coast map, here what you can see is that there's absolutely no overlap at all, with one exception maybe on chromosome 20. But even these confidence intervals, they don't overlap, they abut each other. So to us, this was incredibly remarkable because this suggests that even though the tail morphologies are almost identical on the East and West Coast, the underlying genes responsible for those differences in tail length seem to be completely independent. One thing that you may notice when you look at the West Coast QTL is that there's sort of this funky QTL here shown on chromosome 15, which shows a really strong association with tail length. But its shape is funny because the association is not only high, but it is consistent across about half of the chromosome. We've dug into this further, and it turns out that there's a large chromosomal inversion, about 40 megabases in length, that is not only associated with longer tails in the West Coast mice, but also is associated with darker coat color. So it seems like this inversion is carrying 2 mutations which contribute to adaptation to forest life. So you can read more about this in a bioRxiv preprint. But the main thing I wanted to sort of conclude here is that there's this remarkable convergence in morphology is not due to the same gene. Okay, so let's return now back to the East Coast populations, and I'm going to focus on these for the rest of the talk. And in particular, I'm going to focus on the trait, the number of vertebrae, and I'm going to focus on one particular QTL here shown on chromosome 8, because it's the strongest QTL and it's the one that's most repeatable in our data. Okay, as you can notice, this significance region shaded here in purple is actually pretty large, not surprising for a QTL study of this size. But we asked, how do we narrow in on genes that are in that QTL region? And so one of the approaches we decided to take was to do RNA-seq of early and late tail development. So we know the number of tail vertebrae is a trait that's established by birth. So you can see here, forest mice when they're born already have around 27 or 28, and prairie mice have approximately 24. So what this means is there's something interesting going on during embryogenesis that establishes this trait divergence. So we did bulk RNA-seq at 2 time points that are relevant for tail development and asked, are there any genes that are differentially expressed between forest and prairie mice? And for those of you who've done RNA-seq, it's not surprising that not only did we find a few genes, but we found literally thousands of genes that are differentially expressed between these 2 ecotypes. And this is notable in part because, remember, these populations are separated by approximately 10,000 years, yet we see this amount of divergence. So this approach alone isn't going to point us to a handful of candidate genes that we can functionally manipulate, but the idea was to ask, of the genes in this QTL interval, remember this interval is large, it contains approximately 140 protein coding genes, which of those genes also show differential expression? So as I mentioned, there's thousands of genes that are differentially expressed, but when you think about the overlap between these 2 approaches, there are about 25. Again, this is still a lot of genes to work with, so we took one more filtering approach to prioritize candidate genes. And that is, we looked at genes that are known to affect tail phenotype in laboratory populations of mice. There are a lot of these actually, but only 3 of them are differentially expressed in our QTL region. So what are these 3 genes? Perhaps not surprisingly, 2 of them are Hox genes, and one of them is a gene called SP5, which is involved in early embryonic patterning and Wnt signaling. Now we started to dig into these genes, but one gene in particular, HOXD13, caught our attention. So let me explain why. So as I'm sure you're all aware, one of the coolest things about Hox genes is that they're collinear. That is, their organization on a chromosome mirrors where they're expressed in a developing embryo. So HOX13 genes are the last genes in the cluster, and they're expressed in the developing tail bud at the end of the embryo. So they're kind of expressed in a relevant place. In addition, there's been some beautiful developmental genetic studies done by Olivier Porquier, the Deschamps lab, that showed when you increase HOXD13 expression, it's a signal to stop elongating growth. So you can think of HOXD13 as a stop signal, and that when that's expressed, you end up getting mice that have relatively short tails. So with that in mind, we looked at this data, and at least it's consistent with that in the sense that the short-tailed prairie mice have high levels of HOX earlier in tail development, whereas we see an increase in HOXD13 expression later in the long-tailed forest mice. Moreover, when we looked at allele-specific HOXD13 expression in these F1 hybrids, you can see it largely recapitulates the difference in expression we see between the parents at both time points, and suggests that there are cis-acting mutations that are responsible for the differences in HOXD13 expression, and is consistent with this gene being under our QTLP. Okay. But all of these experiments sort of have us to HOXD13 as an exciting candidate gene. We're currently doing a number of experiments to show that manipulation of the expression of that gene actually causes the difference in tail phenotypes. But while we were doing that, we wanted to ask another how question, and that is, how during development do you get differences in tail length? So this gets to your question. So as I mentioned, all the action happens during embryogenesis, and we know a lot about tail development, again, from studies in mouse and chicken. So there's a really important time point about E10 through E15 in which the tail really develops in mice. And I'm going to give you a brief, very simple overview of what happens during that time point using this chicken embryo as a model. So caudal vertebrae develop from somites, and those somites are essentially sliced in a regular clock-like fashion from this material here, the presemitic mesoderm. I like to think of a presemitic mesoderm as a loaf of bread, and you're slicing that at irregular intervals. So you get these slices, which then are your somites that then form these caudal vertebrae. So if you start thinking about how you might get more caudal vertebrae, there are again two sort of simple hypotheses that we started with. The first one is you can start with the same loaf of bread between prairie and forest mice, but you can just slice that in thinner slices and maybe at a faster rate. And that would result in more somites, but also the somites on average should be smaller. Alternatively, you could start with two different sizes or lengths of bread, and you could keep the slicing the same. So in this case, the somite size would be the same. So we did a number of experiments. I'm just going to summarize years of work in two simple slides. The first thing is to say we didn't see any difference in somite size. So here's the length of the last form somite. Here are the number of, think of this as developmental time. There was no difference between forest and prairie mice. However, we did see a difference in the length of the pre-Semitic mesoderm when we measured this in fixed embryos, and it was in the direction we expected. That is, forest mice have larger pre-Semitic mesoderm. So this, in addition to a lot of other experiments, including explant experiments and so forth that we've done, really suggests that most likely it's differences in the size of the pre-Semitic mesoderm that's the developmental mechanism that's contributing to these differences in vertebrae number. Okay. So from a developmental perspective, we've been focusing on the pre-Semitic mesoderm. From a genetic perspective, we've been focusing on our favorite candidate gene, HOXD13. And the question was, are these two connected? So to address that question, what we looked at were the progenitor cells that are responsible for giving rise to the PSM. And these are these neural mesodermal progenitor cells that live within the chondroneural hinge that we can identify and count when we double stain with these two markers. And when we did that and compared forest and prairie mice, we saw a difference with forest mice having more of these neural mesodermal progenitor cells, which contributes to the differences in pre-Semitic size. Moreover, HOXD13 plays a role in the generation and proliferation of these NMP cells, thereby providing a nice plausible link between our candidate gene and our developmental mechanism. Okay, so now let me step back and summarize what I told you. So this study started off with this natural history observation over a century ago that there are these two ecotypes of mice, forest and prairie form. I showed you that there's a difference between these two ecotypes in their climbing performance, with forest mice performing better. From a morphological perspective, I showed you that the tails are different, that with forest mice having longer tails. This is because they have both more vertebrae and longer vertebrae. And we think that Hox genes acting on NMP cells give rise to a bigger pre-Semitic mesoderm, which affects vertebrae number, which gives rise to differences in tail length. But what's missing from this is that tail length actually affects climbing performance. I showed you that it's correlated with. So in the last data slide, I want to show you two more slides that makes this final link. So remember, we generated this large population of F2 recombinant hybrids, all of which have different tail lengths and different combinations of genes. Because their genomes are shuffled, this allows us to dissociate within these mice tail length from other traits like whisker length and foot size. And so by measuring their tail morphology and then also running all of these mice in our climbing assay, we can ask, does tail alone have an effect on climbing performance? So indeed it does. So there's, as you can see here in this canonical correlation analysis, there's a really strong and positive correlation between morphology and performance. But what's most interesting, the loadings on these axes, and it's mice that are small, that have big feet, but also independently have long tails that spend more time upright. When they're upright, move at a higher speed and spend less time upside down, thereby suggesting that tails, independent of these other morphological traits, actually affect performance. So today, what I hope to convince you of is that starting at this morphological variation, we're getting a handle on the genes, how they interact through development to produce variation in trait, and what the consequences then are on performance. In other words, understanding both the evolutionary processes and the molecular mechanism driving trait variation. So with that, let me thank Evan and Emily again, who were really the main drivers of this work. Work on the West Coast mice was done in part by Olivia and Brock, We had a team of research technicians, wonderful undergraduates, and 2 new postdocs that are working on different aspects of climbing behavior, both from a developmental and from a neurobiological perspective. And with that, I'm happy to take any questions.